Stem cells. Stem cells are mother cells that have the potential to become any type of cell in the human body. One of the main characteristics of stem cells is their ability to self-renew or multiply while maintaining the potential to develop into many other types of cells. Stem cells can become cells of the blood, heart, bone, skin, muscles, the brain, etc. There are different sources of stem cells, but all types of stem cells have the same capacity to develop into multiple types of cells. They are valuable as research tools and might, in the future, be used to treat a wide range of diseases. This is a debatable topic that has sparked much controversy. Different types of stem cells. Scientists first studied the potential of stem cells in mouse embryos over 2 decades ago. Over years of research, they discovered the properties of these stem cells in 1998. They found methods to isolate stem cells from human embryos and grow the cells in a laboratory. Now, there are 3 types of main cells. The first one, we start out with pluripotent cells. Pluripotent cells possess the capacity to divide for long periods and retain their ability to make all cell types within the organism. The best type of pluripotent stem cell is the one present in embryos that helps babies grow within the womb. These are termed embryonic stem cells. These cells are the most controversial. Fetal cells. Fetal cells are obtained from tissues of developing human fetus. These cells have some characteristics of tissues that they are taken from. For example, those taken from fetal cells can make only muscle cells. The difference between embryonic stem cells and fetal stem cells is that the fetal stem cells have matured part of the way to becoming mature adult cells. The third type, adult stem cells. These are obtained from tissues of the adult body. The most commonly used example is bone marrow. Bone marrow is a rich resource of stem cells that can be used to treat some blood diseases and cancers. Adult stem cells are the reserve supply of cells that can multiply when needed for the repair of adult organs and tissues. Now, the pros and cons of stem cell research. Stem cell research is a very controversial subject, and the pros and cons of stem cell research come to the surface when we examine these fetal and embryonic cells. These cells are extracted directly from an embryo before the embryo cells begin to differentiate. At this stage, the embryo is referred to as a blastocyst. There are about 100 cells in the blastocyst, a large percentage of which are stem cells, and can be kept alive indefinitely. The replicating set of stem cells from a single blastocyst is called a stem cell line, because the genetic material all comes from some fertilized human egg that started it. President Bush authorized this, federal funding for research on the 15 stem cell lines, available in August of 2001. Other stem cell lines are also available for research, but without coveted assistance of federal funding. Against this, the embryonic research advocates argue that the tiny blastocyst has no human features. Further, new stem cell lines already exist due to the common practice of in vitro fertilization. Research advocates conclude that many fertilized human cells have already been banked but are not made available for human research. Now, this is one of the biggest problems. And now we move on to the purpose of the brain today. Brain injury and what it is. Brain injury is a type of neurotrauma caused by external or internal forces that disrupt the functioning of a brain. BI can result in a range of physical, cognitive, emotional, and or behavioral effects that can have outcomes varying from recovery to disability and even death. Unlike standard treatments, stem cell regimen is different by the way it targets to repair the brain tissue, damage itself, and recover functions. Stem cell treatment focuses on the root of the problem and not necessarily the symptoms. Cell death occurs when cells are injured. However, these dead cells are surrounded by damaged and healthy cells. Stem cells have the potential to stimulate the healing of these injured cells by the secretion of cytokines, such as nerve growth factor, to promote the body's self-repair mechanisms. The objective of the treatment is to repair the injured cell area around the lesion itself. This will lead to improved symptoms in physique, movement, and intelligence. The majority of patients show improvements right after the first or second transplant. They continue to improve for about 6 months to 1 year before the final results settle in. For brain injury patients, the achieved results are permanent. Researchers encourage stem cell research for the study of these neural stem cells as potential transplantable tissue for the repair of injury, such as that sustained during traumatic brain injury or even stroke, for the repair of pathological processes such as those seen in the neurogenetic diseases, or for repair of neurodegenerative processes such as those seen in Parkinson's or Alzheimer's diseases. In addition, the stem cells should be used for stem cell research aimed at the detailed study of mechanisms of neural differentiation and transdifferentiation, the genetic and environmental signals that direct into specialization of the cells and particular cell types. and as in vitro models of diseases such as autism and Fragile X syndrome. Now, examples of stem cell research and the brain. Johns Hopkins researchers have found that stem cells from a patient's own fat may be able to deliver new treatments directly into the brain after removal of the most common and aggressive form of brain tumor. These stem cells are called mesenchymal stem cells, or MSCs. Now, MSCs have the ability to seek out damaged cells, investigators say, such as those involved in cancer, and may provide clinicians a new tool for accessing difficult-to-reach areas of the brain, where cancer cells can hide and proliferate. Harvesting these stem cells from fat is invasive and less expensive than getting them from bone marrow, or a more commonly studied method. Currently, standard treatments for glioblastoma, the most common and aggressive form of brain tumor, are chemotherapy, radiation, and even surgery. But even a combination of all three rarely leads to more than 18 months of survival after diagnosis. Glioblastoma's tumor cells are particularly nimble, migrating across the entire brain and establishing new tumors. This migratory capability is thought to be the reason for the low cure rate of this tumor type. Stem cell research continues to grow and will be a major impact on the quality of life all across the globe. Not only will it have a major impact on brain diseases, but also on diseases all over the human body. For those reasons, I thank you for my presentation today on stem cell research and biomedical engineering and the brain.